perché sono un paio di notti onestamente che lavoro fino a tardi e quindi sono arrivato un po' ai minimi termini. Poi queste riunioni una sera abbiamo la riunione del giovedì noi per organizzare la conferenza di neuroingegneria che ci massacra, anzi la prossima la salto, Calogero, guarda.
Sì. Siamo in diretta a YouTube, questo è giusto per informare.
Magari se me lo dicevi prima, non la adoro.
Però il professore
ho detto che sono stanco.
Grazie Simone, grazie molte.
Grazie mille, buonasera. Ho detto che queste riunioni delle 10 di sera, cioè, abbiamo il giovedì quella per il congresso ed è faticosa. Professoressa Cirelli, buonasera.
Buonasera Chiara.
Grazie ancora. Allora, dicevo Ugo, a nome dell'istituto l'introduzione e la conclusione la fa Calogero come collega che ha organizzato per noi tutto quanto. Lascio a lui l'onore di rappresentare il nostro istituto.
Va bene.
Io però sarò qui.
Ciao Chiara.
Ciao.
Nella speranza, professoressa, di averla con noi, dicevamo per email, diciamo, davvero non virtualmente appena possibile.
Per usare un termine molto pisano, di ciccia, come si dice.
Ecco,
di ciccia.
Bravo. Ecco, bravo, guarda Ugo, si va a mangiare alla grotta come tradizione, si mangia un po' di ciccia, un po' di vino buono, un gotto di vino.
Io mi ricordo, appunto, ne parlavamo quando ero allievo al primo anno o al secondo, non mi ricordo nel 2002 nel 2003, che proprio fu un bellissimo seminario di Chiara Cirelli e Giulio Tononi insieme, che devo dire per me furono di grande ispirazione nel mio percorso, diciamo, di studente al tempo, poi il percorso accademico successivo. Ho fatto cose diverse, però ecco, le tematiche delle neuroscienze sono state importanti, diciamo, lo stimolo che ho ricevuto è stato importante anche da quel seminario. Poi magari sono quei 4, 5, 10 seminari che hai seguito, no, in qualche anno che ti hanno condizionato un po'. Devo dire che quello lo ricordo come una delle esperienze che ha condizionato lo sviluppo del mio percorso. Quindi spero che lo stesso possa fare per chi è connesso.
Per gli altri studenti, certo, per quelli che...
Per il commento, e come abbiamo appurato poche ore fa con Ugo, quello che ho detto allora, che il sonno è importante, sembra essere ancora vero, in base ai suoi studi recenti, in the field, e quindi...
In the middle of the sea.
In the sea, exactly.
Scusate, approfitto per fare una domanda ad Ugo. A un certo punto tu fosti citato in un senato accademico o inaugurazione dell'anno accademico come un ragazzino che a 6 anni, questa storia non so se la sa perché è molto bellina, professoressa, praticamente Ugo dice che la mamma a 6 anni gli disse: Ugo, vai a fare il riposino pomeridiano perché il sonno fa bene. E Ugo guardò la mamma e disse: mamma, non ci credo, non sono convinto che sia così. Questo me l'hanno raccontata, non so se sia vera.
Allora, temo che l'autrice di questa cosa sia la Laura Liberti, Calogero, tu che sei la memoria storica e preservi ogni sinapsi. La Laura Liberti mi è stata raccontata così, io non me la ricordo di prima mano, ma mi è stata raccontata, ma credo che fosse semplicemente che non volevo andare a dormire, quindi trovavo delle scuse. E quindi, perché devo andare a dormire?
La mamma saggia?
E la mamma saggia?
Può entrare nella narrazione che uno costruisce di te, comunque.
Esatto. Però insomma io mi opponevo al sonno e mia madre mi disse che dovevo andare a dormire perché sì. Cosa che mi sembra una spiegazione un po' approssimativa, insomma. Diciamo.
Non rimanesti cioè non ti riusci a convincere fino in fondo, diciamo così.
Esattamente. Allora, vedo che ci sono tanti collegati, non so, appunto, considerato l'orario... noi migreremo sull'inglese come abbiamo anticipato a Chiara.
Yes. And also I must say I'm impressed that you do evening seminars for the PhD program. That's very civilized, but
and you know,
demonstrates a lot of commitment. Okay, should I share my screen?
Yes.
Yes, please. Oh,
Ciao, Chiara.
I can see Giuseppe and Marta there.
Wow.
I don't know whether you see them, Chiara.
No. Can you see the screen?
Yes.
Okay, I put full screen. You see it?
Mm-hmm.
And you see my pointer?
Yes. If you don't mind, can we record the lecture?
Sure. I don't mind.
Okay.
Yeah.
Thank you.
No, I don't see the names. Maybe if you can save the names for me at the end, that would be nice.
Yep, we will. Absolutely. So, by the way, in parallel, the Associazione Allievi is streaming the event on YouTube. So we are reaching out. So.
Yes. Well, I'm impressed.
Okay. I got a message from Filippo Bordoni, who is the president of the Alumni Association. I think we can start. What do you think, Calogero, Silvestro? Should we start?
Yes.
Okay. So can you activate your mic and webcam?
I am on, I think.
Yeah, you are, but I think Filippo wanted to welcome you.
Here I am.
Okay, Filippo.
Can you hear me? Okay. I'm sorry, but I was experiencing some troubles, some connection troubles. Sorry,
Filippo. Just one note for those that are streaming in YouTube. Can you press ignore over the warning for the recording so that doesn't overlap? Okay. I asked for the guy that is streaming the presentation on YouTube. Okay.
Yes, it's Simone.
Okay. Thank you. Okay. Good.
Filippo, we can start. So a very brief introduction, and then we will start the seminar. Filippo.
Okay, thank you very much, Ugo. So good evening to everyone. I am Filippo Bordoni, and I'm the current president of the Students Association of Sant'Anna School. The Students Association is just a small association that gathers together PhD students and allievi ordinari, or honor students, that we believe are somewhat the backbone of our academic environment. And we organize cultural events, concerts, and of course seminars like this one we're having tonight. And of course, if you want to keep in touch with us, you can reach us on the social networks. And well, tonight's seminar would not be possible without the effort of the Associazione Ex Allievi, the Alumni Association, and its president Ugo Faraguna, and two brilliant professors from the Biorobotics Institute, which are Calogero Oddo and Silvestro Michela, which I'd like to thank for giving us this opportunity. And last but not least, of course, I would like to welcome our prestigious guest, Dr. Chiara Cirelli, which, by the way, is also a former medicine student here at Sant'Anna School. And I believe this will be a great opportunity to learn something new about sleep, but also a way to keep in touch with former students of Sant'Anna School, which is a part of our mission. So thank you for accepting our invitation, and have a nice evening.
Thank you very much for the nice invitation. My name is Chiara, and yes, I have been at the Scuola for many wonderful years. So I'm really grateful for this opportunity to present what I have been doing since I left the Scuola. And I hope this is also an opportunity perhaps to establish new collaborations for the future.
Just if Calogero wants to add a couple of words, and then I will introduce very briefly your biography, Chiara. So sorry,
I just want to check. Okay.
It Perfect.
Thank you.
Calogero.
Thank you, Ugo. Thank you, Ugo, for let's say being the link with Professor Chiara Cirelli. We are glad to have so highly distinguished lecture tonight in our doctoral school in collaboration with the Alumni Association and the Students Honors College Association of the Scuola. Our PhD, the PhD program in biorobotics, is among the largest doctoral schools worldwide in robotics and bioengineering. And we are organizing a series of seminars as a part of the educational program for our PhD students. This is organized in transversal cycles of seminars on cross-disciplinary topics and on vertical seminars on, let's say, specific topics that are related to the cultural scientific areas of our institute. In this specific seminar that we host in the cycle of seminar on neuroscience robotics, together with my colleague, Professor Silvestro Chiara, this will open the series on neuroscience robotics, and then we will host other distinguished lectures. I'm very glad that we have this collaboration. We are not doing for all seminars, but for some selected seminars that are meaningful, we are having this collaboration with the Alumni Association and with the Honors College Students Association. By the way, I am a co-founder of the Honors College Association. We were 17 students co-founding the Honors Student Association. I was one of them. Ugo, for sure, remembers those periods we were together at the school as Honors College students. And I am also, let's say, part of the Honors of the Alumni Association. So I'm very much linked to all the parties that co-organize tonight the initiative. Okay, so let me thank again Professor Chiara Cirelli. Let me also remark that soon we will have a master's student, one of our Honors College students, Giuseppe, that will spend a visiting period in Madison in the lab directed by Chiara Cirelli and Giulio Tononi. And I'm sure that this will be a first possibility to establish further links between our doctoral school and our academic school and the one of Professor Chiara Cirelli and Giulio Tononi. To conclude, let me remark that I was really, really inspired by one of the talks given by Chiara Cirelli and Giulio Tononi when I was a young student. So I really hope, and I'm sure it will, that this will be a real opportunity to define the track for a young generation that are now connected in this sector. Thanks again for being here.
Thank you, Calogero. And just a couple of words to introduce Chiara. I would define Chiara as a golden alumna of Scuola Sant'Anna for many reasons. She's been both allieva ordinaria and allieva perfezionanda. So this is a golden match. And then she's golden in the sense that she shaped the field of basic sleep research, in which she is a world-renowned authority. After receiving her medical degree from the University of Pisa and her PhD in neuroscience under the mentorship of Professor Pompeiano, she has been studying the function of sleep. She moved to the Neuroscience Institute in San Diego, directed by the Nobel Prize Gerhard Morris Edelman. And then since 2001, she is a faculty, now full professor at the Department of Psychiatry, University of Wisconsin, Madison. She will describe in her talk an impressive amount of different techniques that she adopted in order to answer to the basic question of what the function of sleep is and what are the effects of sleep deprivation. In 2017, together with Giulio Tononi, also an alumnus of Scuola Sant'Anna, she got the prestigious Farrell Prize in Sleep Medicine from Harvard Medical School. And in 2018, she was awarded the Outstanding Scientific Achievement Award from the Sleep Research Society. As in the Italian academic world, we are obsessed with the bibliometric indexes. Just to give a hint of Chiara's impact on the field, her work has been cited overall more than 22,000 times. Her H index is 70, and she has been included by a systematic analysis from PLOS Biology in the top 2% list of most productive scientists in any field in 2019. Finally, and it's a finally, and this is rather relevant for the alumni and the allievi, the doors of Chiara's lab have been open over the past years to several allievi, including myself. In Chiara's lab in Madison, Wisconsin, I had the privilege of spending 7 years, unforgettable years, as PhD student first and as postdoc later. With warm and deep gratitude, I leave the virtual floor to Chiara. Thank you.
Thank you very much, Ugo, for these very nice words. I must say I feel very old, but I hope you can, from my talk, gather the impression that I still very much involved. I still believe very much that there is a lot that we need to discover about sleep. And really, sleep has always been my passion from the very beginning when I started as a medical student in Pisa. So the work that I'm going to present is done in collaboration with another ex-alumno della scuola, Giulio Tononi. Is done now at the Wisconsin Institute for Sleep and Consciousness, and is supported by NIH and the DoD. And I have no conflict of interest to declare. So as Ugo mentioned, we are trying to understand why we need to sleep and why we spend overall a third of our life in this behavioral state. And this is the case also for all the animal species that have been carefully studied so far, 230 or so. And in this slide, you see on top some of the latest additions to this list. For instance, here is on the top left the jellyfish Cassiopeia, and on the right the flatworm, and here the hydra. So in one sense, you can say that asking why we need to sleep is kind of maybe unwarranted, that after all, of course, we know why we do need to sleep. Because if we don't, we feel miserably the next day. Everything is extremely difficult, everything is gloomy and hard to achieve, which is absolutely the case. And there is a lot of evidence, consistent, converging evidence in animals and especially in humans that indeed sleep loss affects very broadly performance and cognitive function. In fact, it's quite difficult to find one aspect of cognition that is not negatively affected by sleep deprivation, from attention, specifically sustained attention, learning and memory, including working memory, the ability to integrate a lot of information, to switch strategy, to speak clearly, to assess risk or inhibit behavior. Everything is affected. And I'm sure many of you know humor even is affected. And even sometimes when you then test subjects in tasks, and you may find that the overall performance after short deprivation is not affected on average, the subjects will tell you that they felt miserably and they felt that they have to use so much more effort and energy to get to the same end result. So this concept of energy allocation is quite new in the field, but there is a lot of converging evidence that indeed this is what happens with sleep deprivation. Now, it's not just that if you are sleep deprived, you have all this impairment. There is also a lot of evidence over the last decades that then once you have this impairment, you can recover from that only through sleep. Just quiet wake or rest, meaning you close your eyes but you are awake, doesn't do it. And here you see on the right, these just 3 studies that are, 2 of them are very recent, making this point and showing formally that indeed rest, quiet wake is not sufficient to recover. You need a nap or a long sleep. So, but the question is, we know that this is the case, but why? What is that sleep affords to the brain that cannot be achieved during wake? That's the question that we are trying to address. And we think that to try to solve this riddle, you really have to start from the very same definition of sleep, the feature of sleep that distinguishes sleep from any other behavioral state, again, including quiet wake. And that's what is depicted here beautifully by this painting by Rousseau. When we are asleep, we are in a state of partial sensory motor disconnection, which means that we lose the ability to promptly respond to a stimulus and potentially to avoid a very dangerous situation. So going back to what I showed you before about the fact that we spend a third of our life asleep and all animals do the same, then you have to ask, there must be at least one very good essential reason why we do that and we take this risk. Because if not, evolution, we reason, should have found a way to carry out the same function during wake, when we are still able to respond to the danger. And we think that function that is essential and can only be carried out very effectively in sleep but not in wake is to maintain synaptic homeostasis. And that's what I'm going to tell you about. I'm going to tell you the rationale for this hypothesis and the evidence that we have so far, including at the end some unpublished data that we just have completed. So you know that synapses are the way neurons in the brain communicate to each other. What you see here on the right is the schematic of the paradigmatic type of chemical synapse in our brain, in a mammalian brain. These are excitatory, glutamatergic synapses. There is the input neuron and the axon of that neuron here ending in a button. The green vesicles contain the neurotransmitter, the glutamate, that is being released, sending the signal to the target neuron. In this enlargement, the spine, the head of the spine, which represents the post-synapse. So this part is the pre-synapse, and this part is the post-synapse. Now you see here, for instance, that's a 2D image taken with an electron microscope of one of these synapses in a mouse brain. And again, you see this enlargement of the spine head, and here in front, the button with the glutamate vesicles. Now, why would be maintaining this synaptic homeostasis so important? Well, first of all, again, synapses are at the basis of how the brain works, and there are many of them. This is an estimate just for the human neocortex that there are 100 trillion synapses. Second point, synaptic transmission in general is extremely energetically expensive. There are different calculations, but overall, everybody agrees that if you put together the energy needed for synaptic transmission, for emitting the signal, which is the action potential, and then to rebalance the ionic currents, related to that signaling, you basically are spending 60, 70, 80% of the total amount of energy in the brain. And as you know, the brain is already using a lot of energy. So that's why this activity has to be kept under very tight control because it's so expensive. Now, here is a schematic of the general idea of this hypothesis that we have been testing. You see here in this, the 24-hour cycle, the light phase, 15, 16 hours for us, is the wake day. During the day, and that's a fundamental concept in this hypothesis, is that there is always learning, what we call ongoing learning. It's not just when you go to a class or you go to a lecture and hopefully remember some of what I'm going to tell you, but just the fact that you are awake, that you are exposed to the environment, and the environment is always changing, forces your brain and the synapses to be modified to adapt to that environment. That's the learning that is happening all the time. And the end result of this, by the end of the day, we think, is a net increase in synaptic strength. So overall, in many brain circuits, and that's what you see here in these red lines, in this sagittal view of the human brain, there is a strengthening, an overall increase in the efficacy of these synapses. Because of that, we think what sleep does, and is absolutely essential, is that it produces then a renormalization, an overall net decrease in synaptic efficacy in many, if not most of all circuits, which is what you see here in green on top of the slide. And I will try to tell you at the end how we think this is happening. Is a process that we call smart down selection, because he has to be selective and he has to be smart to be able to account for all the beneficial effects of sleep on performance, which are many different ones. It's not just that when we wake up, we are refreshed and ready to learn again, but we also remember some of what we learned the previous day. We have integrated that information with all our previous body of knowledge. And very importantly, actually, we have forgotten many things because many details are irrelevant in the long run. So whatever sleep does, we need to be able to account for all these beneficial effects that have been shown be carried out by sleep at the behavioral level. So these are the 5 points basically at the basis of this hypothesis, the rationale for this hypothesis, which I want to go in some detail because I think it's very important that you get this as the gist of my lecture, independent of the minute details, that sometimes, at least for some of you, may be a little bit obscure in terms of molecular biology and etc. So the first point, as I mentioned, is that learning must occur when you are awake, exposed to the environment. It should be mainly through the strengthening of synapses, which then creates the need for a renormalization. And that we think happens mainly, not exclusively, but mainly during sleep. And again, we think that is this process of down selection that accounts for all the benefits of sleep at the systems levels, the ones that I just mentioned, but also at the cellular level, because at the end, through sleep, through the renormalization, you save energy. Now, learning must occur in wake. Again, it's only when I'm awake that I'm confronting the environment and I need to learn about the causal structure of the environment. And this is ongoing learning. This morning, I took a walk in the woods around where I live, and I have images of what I saw. I also was thinking about, giving these lectures, so remembering about Ugo, remembering about the scuola, etc. All this is laying down many memories, many traces in my brain in the short run. And you would be surprised, but you can do this experiment. If you try to remember everything that you have done today, you will be perhaps surprised at how good you are in the short run, again, in remembering many, many details that may be in the long run irrelevant. This is actually has been tested several times. This is just one classical experiment in which subjects were exposed in 5 hours or so to 2,500 different objects. And then immediately after that, they were asked whether or not just they saw the object, but about specific details of the object. And what you see on the right basically is their performance. And the take-home messages here is 90%. So they were very good, again, in the short run, in remembering all these details. So our brain is extremely good in laying down memory traces. The learning should occur through strengthening of synapses. And you can ask, why would that be the case? Well, first of all, the brain, the chemicals that are present in our brain when we are awake are all very conducive to the strengthening of synapses rather than the weakening. You see here this image that is showing one system, the noradrenergic system of the locus coeruleus. And what this is meant to convey is that this system, like many other arousal systems, which are groups of neurons that we know are very important for us to stay awake, all those systems are much more active when we are awake than when we are asleep. And the neurotransmitters, noradrenaline, acetylcholine, orexin, dopamine, released by these systems facilitate and promote synaptic strengthening rather than weakening. Again, this is not absolute. This is not to say that you cannot have some synaptic depression during wake, but overall, the environment in the brain when we are awake is much more conducive to potentiation than depression. But, and there are evidence, so you can formally, of course, test these. And these images, again, capture some examples of learning, skill learning, procedural learning, declarative learning, fear conditioning, in which it has been properly shown that indeed the circuits involved in this learning are undergoing synaptic potentiation. But you can ask, but why would it be? So if I have a neural network in a baseline condition, I can implement, in theory, definitely you can implement learning through rules of weakening, of depression, rather than potentiation. And so why would it be instead that in us, in animals in general, we go through strengthening? Well, consider that here neurons in our brain, the great majority of neurons, are not directly connected to the environment. They are deep into the brain, and they know nothing about what's happening outside in the external world. The only thing that they know depends on the input that they receive. So these axons synapsing here on the dendritic trees of the target neuron. So that's what they know. And based on these inputs, the only thing at the end that they have to decide, despite being incredibly complex in terms of morphology, molecular biology, etc., at the end, what they have to do is either fire or not fire. So emit these action potentials or not. But it seems a very simple decision, but it actually is not a free decision, meaning there are huge neurobiological constraints to this decision. And the main constraint, again, is what I mentioned before. The firing is much more expensive than non-firing. What you see on top here for the CA1, which region of the hippocampus, but this applies to many regions in the cortex, in rodents, in monkeys. It's a very general finding. What you see here is the firing rate of neurons on average, And you see this is the linear scale and this is the logarithmic scale. So we see the distribution is log normal, which means the great majority of neurons fire very little at 1 Hz, so 1 spike per second, 2 spikes per second. Very few of them have high firing. And again, that's because firing is very expensive. Not only that, but we know, and by now this is very old, but we have a lot of data from new, much easier to collect now data in humans with intracranial recordings, that neurons are also very selective and they only respond consistently, reliably to specific stimuli. So here is one neuron, number 3, presented, each row is one trial, to a series of images, and you see consistently this neuron is only responding to this image and not to the other images. So neurons stay quiet usually, and they only fire when they see some preferred stimulus, and very selectively. So let's go back to this neuron deep in the brain. So what he knows that something is important if he gets an increase in its input firing, especially if it's not just one synapse that gets activated, but if it's more than one, more or less simultaneously, what has been called suspicious coincidence. Then he decides that then he better fires because there is something important. And however, because of what I told you, firing means he's using a lot of energy. And so better be sure that this effort, this price that he pays in terms of energy, has a good consequence, which means that the signal gets transmitted to the target neuron. And to be sure that that's the case, it means that the signal, the synapse, the contact here to the target neurons have to be strengthened, not weakened, because if you depress these connections, of course, then the signal will die off and the signal will not percolate in the chain inside the brain. This is the fundamental reason why you want the system to work through strengthening and potentiation rather than depression. This is not controversial. I think, in most neural networks, that's the way the rules of learning are implemented. And that's also why, of course, you get into this issue of the need for renormalization very soon. And there are many, many studies here. I'm just showing some of them in which it's obvious that if you don't implement renormalization weakening rules, there is runaway potentiation very rapidly. All synapses will get very strong, very saturated. There will be no more learning, and there will be usually epileptic kind of activity. So that's also very established. But what is controversial, and that's where our hypothesis, I think, still remains debated, and many people don't agree, is that in most cases, people think that the brain is smart enough to be able to maintain this balance between strengthening and weakening at any given time. So let's say now, hopefully, you are strengthening some connections related to what I'm telling you. Yet, simultaneously, you are weakening other circuits, other connections. Instead, we think this is not what happens. There is overall a balance that is maintained across at least 24 hours, the sleep-wake cycle, and maybe even longer than that. But within these 24 hours, there is a time wake in which there is a net increase in synaptic strength, again, facilitated by the arousal systems, and vice versa, a time sleep when the opposite happens. In sleep, the low levels of all the arousal systems are promoting weakening. Crucial, you have sensory disconnection. So we are back to the fundamental feature that I mentioned in the beginning. When we are asleep, we are no longer slave of the here and now of the environment. We are not asked to adapt to the environment. we are detached, yet neurons are active, not as active as in wake, but pretty active. And that's actually one of the most important findings overall in the entire sleep research over the last century. Neurons not only are active, but they are more or less active all simultaneously, on and off, in this pattern of activity that creates the slow waves of sleep, which creates a very good condition to do what we call comprehensive sampling. So neurons get activated, the memory traces get activated, and the brain can decide in a smart way to down select and renormalize some of these traces and not others. Now, I understand that this sounds like magic right now. I hope by the end I can show you at least some of the new evidence that we are start collecting as a field to explain how this can actually happen. So at the end of the down selection, as I said, through this renormalization, we should be able to account for all the system level advantages in terms of learning, memory, gist extraction, and the cellular level benefits. The cellular is, again, because if you overall weaken many synapses, by when you wake up, your brain is literally leaner, requires less energy. When synapses are very strong, they require a lot of stuff, lipids, proteins that need to be produced by the neuron and shipped away to the synapses that very often are very farther away from the cell body. All these needs are reduced if you have weaker synapses. And of course, the renormalization then brings the network away from saturation, so it increases the signal-to-noise ratio. Now, this is the gist of the entire hypothesis. And I think the general idea, we learn and we potentiate in wake, and then we need to renormalize, is simple enough. What has not been so simple has been then to test properly this hypothesis. And the reason is because there is no single measure of synaptic strength that you can use, especially if you want to do it in vivo in a freely moving animal. And that's why really this journey is at least 20-year long. And what we have been trying to do, and is ongoing effort, has been to try to use as many measures of synaptic strength as possible. Some are better than others, some are more direct than others, in different species, literally as you see here, from flies to humans, using also network and computer simulations. Flies, because our assumption is that if this synaptic renormalization is really such a fundamental, essential function of sleep, it has to be conserved through phylogeny. It cannot be just through perhaps in mice and not in flies on humans. Now, very briefly, I'll just give you a sense of how we are trying to do that. For instance, one way that you can assess synaptic efficacy and strength is literally by stimulating the input neuron, the axon, with a fixed amount of stimulation, and then recording from the target neuron here what is called the monosynaptic response. And the slope of this, the amplitude of the first response, is a measure, if you keep the stimulus the same, is a measure of how strongly A and B are connected. We have done this in many different ways. This is an experiment in humans by using TMS, transcranial magnetic stimulation, in which you can directly stimulate non-invasively the cortex. And what you see here in one experiment is the slope of this response, again for the same type of stimulus. And you see the slope progressively increases with time spent awake. This red bar here is a night of sleep deprivation. So it peaks after sleep deprivation. And then finally, after subjects were allowed to sleep, here the blue bar, then it goes back. You can do the same with electrical stimulation in animals, for instance. This has been done several times. Now, some of the studies, I'm just giving you here the references, but I've also tried to assess what happens to the network when you get, let's say, to this point, when we know many synapses are potentiated. So the question was, can you still learn when you are here? And some studies have shown that actually you have much harder time to learn because presumably the network is closer to saturation. The evidence is for, let's say, 1 night of sleep deprivation in people, or even just 6-8 hours of sleep deprivation in rodents. And the point that I want to make here is that it's just not, it seems that, it's a theoretical problem that, well, eventually the network will saturate. It seems that the brain operates on a range that is not very far from saturation. And that's basically probably why we need to sleep every night. Now, another way that you can test is in vitro, actually taking slices of the brain of animals that have been either awake for several hours or asleep. And I won't go into the details of this, but basically in these slices you can just record, you don't stimulate. The strength of the connections among neurons in these slides is reflected in how often you can record the spontaneous release of the glutamate in these neurons. And you can, if the network has been strengthened, the frequency of these events, which are called minis, or the amplitude of them, increases. This is the original study that we did that Ugo Faraguna also was involved in mice and rats after sleep, relative to sleep deprivation, or even just normal wake. And the point was that you see a large increase both in the frequency and in the amplitude of these events. This was in 2010, and now this is just to show you that you need patience, but eventually, if it's true, it should be replicated. And indeed, these are 3 studies, recent studies, all done by different other labs in different, mainly mice, but in different areas of the cortex, all getting to the same point that indeed you see an increase in this marker of synaptic strength after wake or sleep deprivation relative to sleep. Now, you can use proteins or genes, the expression of genes, as a marker, as a proxy of synaptic strength. For instance, in flies, there are very good markers of the pre-synapse. If a synapse is very strong, the proteins expressed here in the axonal button, especially one that is called BRP, are expressed in higher levels. And here you see basically the levels of this BRP protein in a control fly that was allowed to sleep all night, and here a sleep-deprived fly. So clearly, what you see here is basically the entire fly brain. In many brain regions, you see an overall increase in the expression of these synaptic proteins. And again, this is also important because if this idea is correct, it should be that this strengthening is happening in many, many regions and circuits. It's not just one specific network. You can do the same in rodents. There, the best markers that we have actually are in the post-synapse, so in the spine. And those are the receptors for the glutamate, especially the so-called AMPA receptors that are a very diffuse, a very specific kind. And we have used them in our experiments simply because there are hundreds of studies showing that the total amount of these receptors or their phosphorylation level increases when there is synaptic potentiation and decreases when there is synaptic depression. So for us, and indeed at the molecular level, that's probably the best marker that is present in the field to assess synaptic strength. And what you see here, again, without going into the detail, but the black bars are the wake. So if you take a chunk of the rat tissue, the brain, the cortex, or the hippocampus, after wake relative to sleep, and you calculate the total amount of these receptors, there is a 20, 30, 40% increase in their levels, which is again is consistent with an overall increase in synaptic strength across large brain areas. This is a much more recent study done by a different lab, getting to the same point, because here for them 100% is wake, and relative to wake, that's the decrease that they see, 25% in these markers with sleep. Now, again, looking yet for another possibility to test this hypothesis, over the last maybe 6-7 years, we invested in electron microscopy, and specifically in serial block phase scanning electron microscopy. And the reason is because we thought perhaps the most stringent way to test this hypothesis was actually looking at the ultrastructure, at the size of synapses. Because fortunately for us, there is a very good correlation between structural and functional plasticity. So stronger synapses on average are also bigger. But of course, synapses, you see here an example again of one of these post the spine head, and this is the axonal button. They're small. So we're talking about hundreds of nanometers or 1 micron. So you need electron microscopy if you really want to measure their size, and especially if you want them to see a difference across just a few hours of sleep and wake. And so that's the serial block phase electron microscope that we have in the lab. They are literally been running basically for 5 years, day and night. It's a wonderful machine and allows you really automatically to acquire, perhaps overnight or in 18 hours, these stacks of images, in this case of the mouse cortex. Now, all this is nice. The problem is that once you get to the stack, then what you see, these nice colored dendrites and spines, that's actually all done manually. So you need to segment manually the dendrites, the axons, and what we measure at the end is the red area, which is the direct area of contact between the button and the spine, because that's the best established marker, again, at the structural level of synaptic strength. And again, I won't go into the details, but it suffices to say that in 7 years or so, we were able to manage at the end to image 2 areas in the mouse cortex, motor cortex and somatosensory cortex in the superficial layers, as well as a region of the hippocampus in the same animals. And the take-home message is here that indeed, with sleep, after 6-7 hours of sleep, overall synapses get smaller. They literally shrink relative to wake. Now, I will skip this, but I just want to give you the sense. So the first study that we published in 2017, this is all what we were able to reconstruct in 3 groups: spontaneous wake, sleep deprivation, sleep, 4 mice per group, for a total of around 7,000 synapses. It took 5 years, 5 people working full-time to get to this. And of course, we were blind to experimental condition until the end. So this is just to say that this is wonderful. Still, it cannot be, at least for the way we use it, we cannot trust automatic segmentation. So it's extremely time consuming. And that's why so far the evidence that we have been able to collect is only very sparse, just because we don't have the manpower and support to do this. But what you see overall is a decrease with sleep. In mature animals, around 18% on average, the synapses in the cortex are 18% smaller after sleep. And this is the distribution, is again log normal, like the firing rate. So most synapses are small, and all these, which are the majority, 80% or so, the synapses in mouse cortex, get smaller on average by 18% with sleep. The ones that don't seem to change very much are only the very large ones. Now, this is in cortex. I won't go into the details, but as I mentioned, we have done this also in hippocampus. And here is what we reconstructed, again, very, very time consuming. It's a bit more complicated because the distribution of synapses is slightly different, but the take-home message is the same. Both types of synapses go down in sleep versus sleep deprivation. And actually in the hippocampus, there is even not just a decrease in size, but for some synapses, even a decrease in the number of synapses, even if these are quite mature animals. Now, we also have done recently a study in very young pups. And why that? Because, you know, you think that if sleep is important for all this plasticity, etc., that should be especially true during development when there is so much to learn and so much refinement going on. And these pups are 2 week, 13-day old. That's the earliest that we can go and still being able to really define behaviorally whether they sleep or not. And again, we have, in this case, we have only 2 groups, sleep and sleep deprivation, because spontaneously these pups don't stay awake much alone. But again, I won't go into the details, these is the reconstructions, but the take-home message is that indeed there is a decrease with sleep. Actually, if anything, is even larger, is 34% in sleep versus sleep deprivation. And contrary to what I showed you before here, on the right, in which I said, you know, is 18%, is the majority 80% of synapses, but the very large ones are spared. Here in the very young pups, everybody seems to go down. So it really seems a global downscaling by 34%. Now, I want to finish by giving you the new evidence, the new study, and again, just to also give you a sense that we are trying, and every time there is some new way of testing this hypothesis, we try to apply this. And we are far from being complete in this effort, but we try our best. So this is a repeated two-photon imaging study in which we want to see, so far, when I say, you know, the animals are awake, spontaneously awake, or sleep deprived, we give them novel object. But one, perhaps, criticism to our studies was that, well, but you know, these animals are not formally learning anything. They are not doing a task. Again, I go back to the beginning when I say it doesn't matter to us because you are always learning when you are awake. But we wanted to test this. So here the mice is in a task, a difficult task. He has to learn in 1 hour or so to run on top of this complex wheel, which has a irregular rung pattern. So some rungs are missing. So he has to learn to balance, but also to the sequence of the rungs. And there is no pre-training on purpose, so this task is quite difficult. We have shown before that this task benefits from sleep. And we have confirmed this part really looks exactly what we published a few years ago, which is the following. So this is the session 1, the 1 hour of training. And you see 2 groups of mice, but up to here, nothing differs in the 2 groups. So clearly they're learning. By the end of this session, they can manage to stay on the wheel at higher speed. Then you split the 2 mice in 2 groups. Half can sleep for 7 hours, the other half are kept awake. And then you test them again the next day, you put them back on the wheel, and that's what you see. The animals that were allowed to sleep here start performance exactly the same level as they ended session 1. So they have maintained their performance. And instead, the sleep-deprived group, as you see, has degraded performance. So we can say that sleep indeed allows offline consolidation. Now, but this is only the behavioral part of the study. What we want to do in these animals is to look in motor cortex, because we know there is a lot of literature showing that to learn this task, you engage directly the superficial layers of motor cortex. And in these animals, we can see literally these neurons. They are red because through in utero electroporation, we injected actually 2 traces in these animals. The red tracer allows us to track the cell body and all the dendrites and the spines in these neurons, so it's a morphological marker. And then we have the green one, the SEPGLUE1, which is again a marker of synaptic strength because it tells us how much of these excitatory AMPA receptors are there present. This is a very nice marker actually, because it only fluoresces, so it's green only when the receptor is expressed on the surface. So we know it's functionally significant. And so here is what happens to these animals. They are first they are imaged through 2-photon imaging in vivo repeatedly 4 or 5 times, first before sleep and then after sleep. Then they're trained the next day and they're again imaged immediately after learning session 1. Then they're split and then they are imaged again after sleep or sleep deprivation, and then again the next day. And the point here is that finally, because all what I've shown you until now basically was based on comparing electrophysiologically or mainly molecularly or structurally different groups of mice. But here is the same mouse and the same synapse literally that we can follow after sleep, after learning, after sleep deprivation, etc. And what we measure is the green signal, which we measure as normalized difference. So it's the post-session minus pre-session divided by their sum. And this is the overall result. And this is 3 years of work, but at the end, what you measure is this normalized difference. For all these synapses, we have 12 mice, 1,500 or so of these spines. And what you see here, first of all, is the first 2 sessions, so you compare after sleep relative to before sleep. That's with no training. The overall result is a negative ND, which means that overall there is a net decrease in synaptic strength using this new marker, which again is consistent with all what I've shown you before. Here is after learning. So now we are comparing time 0 immediately after session 1 relative to either this or better this time point, which is the same circadian time 24 hours earlier. Here, the ND is positive. As expected, motor learning produces potentiation, so the ND overall is positive. But the most interesting part for us is this. Immediately after learning, if you allow the animals to sleep, at the end, the result is a net decrease, a negative ND. But this does not happen if animals are sleep deprived. And this, again, we have other evidence in flies, for instance, but this is a very important point because you can even imagine, well, once I've potentiated, I've strengthened my synapses here, maybe then they go down in strength no matter what, due to passage of time. Instead, this clearly shows you that you need the sleep again. If you cannot rely simply on wake. Now, this is the summary of what I told you. Really want was to finally be able, perhaps, to answer this question. So when you are at time 0 here, immediately after learning, you have learned to manage the complex will. What happens to the spines, to the synapses that were directly engaged in this learning? And to answer that, we took what we call the learned spines or max spines, meaning the ones that showed the largest increase, the largest potentiation after learning relative to before. And you see here 2 lines because I'm plotting these spines, these synapses, for the 2 groups, the sleep and the sleep-deprived mice. But actually up to here, all mice were not sleep-deprived. They underwent exactly the same condition, okay? But what happens afterwards? Well, quite surprisingly, I must say, these max spines behave in the same way in both groups. They stay up, so they lose a little bit of strength here, but even 24 hours later, they are much stronger than before learning. Okay, so in this case, sleep doesn't really matter per se. However, now let's look at all the other spines, synapses. So these max spines are a small minority, maybe 10% of all the synapses in this motor cortex. 90% do not change because of learning. And here is what happens after session 1. Again, and that's why you get an ND positive in sleep deprivation, they really don't change much. In sleep, they go down. So now if you put those two things together, you get this effect interaction, significant interaction, which means basically the following. So the max spines per se are behaving in the same way after learning, but actually, because of the differential behavior of the other spine, after sleep, the max spine in the sleeping animals are in a better position. They have a relative advantage as opposed to the ones in the sleep-deprived mice, simply because everybody else got downscale, got renormalized. And so if you are back now to what I shown you before, now we have to try to understand why these animals that slept are better off in behavior. And what I've shown you here is that overall, with sleep, they lost synaptic strength. Well, what we see is a negative correlation between the down regulation of spines, of all spines or all the other spines that are the majority, and performance, which means the animals, they are better off when they start session 2, which are the sleeping animals, are the ones that indeed lost synaptic strength. So the summary is the following. So it seems that sleep is promoting offline consolidation in this task, and is doing so by increasing the signal-to-noise ratio. But this happens not because it's the max, not because it's the signal that is increased. It's the noise, it's the other spines that are differentially affected. They go down, they lose, and that's why you increase. So the signal-to-noise ratio increases because of the decrease in noise. And by doing so, you can see how sleep, and this is my last slide, really can get it all, because on one hand you have the behavioral improvement, but at the same time, because of this effect on the noise, which is the majority of spines, you get all this saving in terms of supplies and energy, and of course you move away the network from saturation. So I just want, I know this has been a lot and is quite perhaps detailed. I just want to finish by saying that we know there is a lot of studies going on in different labs trying to understand how can this down selection come that the max spines don't change, and there is the other spines that go down. And there is a lot of studies electrophysiologically or at the molecular level that are showing that, you know, if you use neurons or synapses during wake, you put them in a position to be in a differential advantage relative to the other synapses. And at the molecular level, there are at least 3-4 candidates that we know of now that are targeting synapses that are being used relative to the ones that are not used, and can make this renormalization selective. Thank you for your attention.
Thank you, Chiara. Thank you so much. It's kind of weird not to be able to clap, but unfortunately,
well,
so
yeah, it's kind of surreal, but yeah.
Yeah, but we will repeat it di ciccia, as we mentioned before. So ask Matteo Bucci from Associazione Allievi if it can be so kind to moderate the discussion. Matteo, are you there?
Yes, I'm here. Unfortunately, I can't activate the camera, so I'm here only with my voice. So
you are shy, which is the acronym of Synaptic Homeostasis Hypothesis.
Yeah. So I think that if anyone has a question, can raise his virtual hand, and we can start from that. Thomas, you can speak.
Yes. For your presentation, it was a very, very nice presentation. I would like to know, maybe you can explain if there are some difference in this, we can say, washout of the synapses during the REM phases of the sleep or during the slow wave phases of the sleep.
Yes. Well, the short answer is I have no idea, because we haven't been able to test this directly until now. And so all what I've shown you is the comparison between wake and total sleep. Okay. And there is a difficulty in animals, especially in rodents, because, REM is very limited, and we haven't done any selective deprivation of one state relative to another. So we can only say, which in one sense, if you wish, at the end is what counts, that the end result of total sleep is this. Whether REM does the same job as non-REM, we don't know. It could be the same, it could be different or even opposite. I really don't know. My bet, but again, I have no evidence, is that actually REM does something quite similar in the same direction as non-REM. And the reason is I stressed a lot the fact that, a fundamental factor that helps the weakening is the neuromodulatory milieu. So the fact that noradrenaline is very low, dopamine, orexin is low, etc. And that is shared between non-REM and REM. But again, I have no direct evidence to support my gut feeling.
Okay. Thank you.
Yes, Professor Ugo raised his hand, so you can speak.
Chiara, just a very quick question. So you showed how different methodological approaches have been used to try to tackle these fundamental questions. And it might be a spoiler, but I dare to ask, what do you think should be the next technique to tackle this question? Because with the technological developments, more questions can be answered, and probably we are facing a technological limit so far, and new techniques are available, and your lab is equipped with an enormous amount of different approaches. Imaging has been the last one, but what do you think should come next?
I think one is probably a dream experiment that I would like to perform, or actually maybe somebody else should perform, is actually combining two techniques that are already available, but they are very difficult to combine. One is this kind of repeated two-photon imaging with SEPGLUE1, which is very, very good. Two, because this, again, on one hand, you have synapse resolution, but you don't know what happens to the entire neuron. And the electrophysiology is telling us that, in terms of firing, it counts whether neurons are firing or not during wake and the way they respond to learning. Now, there is a beautiful model of neuroprosthetic learning that many of you know, used very much in San Francisco by Gulati and Ganguly and colleagues, in which they teach, basically they implement learning through direct manipulation of just 2 motor neurons in the primary motor cortex, and they have characterized what happens in the firing of these neurons that they call the causal neurons relative to the non-causal neurons with sleep. If we could combine, in these animals, they use rats usually, but you could do this in utero electroporation in rats and see simultaneously the firing, so more on a network activity, and the synapses in these neurons literally during the development of the learning skill, in this neuroprosthetic learning, and then sleep, that would be very informative. Right now, more than, at least in my view, more than new techniques, we need more evidence in different circuits, in different areas. So, that's the reason why we use it. This skill learning is very well characterized. But what about other forms of learning, other areas? What about the, striatum? What about the cerebellum, which has always been my, dream to study the cerebellum, because in theory, in cerebellum, the rules of plasticity can be quite different. So all these, does it apply to the cerebellum or not? We have no idea.
Thanks, Chiara.
Professor Oddo.
Yes, thank you very much. Opportunity to listen another talk from you in less than other 15 years, because I was
yes, everybody came. I hope so too.
Thanks a lot, really. So I have a question that is connected to what to your last conclusion. So in my non-expert knowledge of sleep mechanism, that the gating areas in the brain to disconnect, let's say, the input of sensory stimuli to higher order regions is in the thalamus. At least there is a role that I've read of the thalamus in this. However, if the gating is actually in the thalamus, and this is the first question, okay? If it's actually there, what happens to the learning in the pre-thalamic regions? As an example, the Kunet nucleus for touch and the Olivary nucleus for audition that are pre-thalamic. And they are second-order neurons, so they are not first-order neurons that we may say they are hardwired somehow. Okay, so they, at least in some modeling, they are exposed to learning as well. So, but if they are not exposed to the gating mechanism of thalamus, if thalamus is the gate, how they can learn?
Yeah, that's a very good question. And again, that's why I go back to the issue that we need to study more circuits. Now, on one hand, well, first of all, the gating, where the gate is exactly in sleep is very controversial. And it may seem to change depending on the sensory modality that you're using. So auditory versus, tactile, or for that matter, olfactory doesn't go through the learning through the thalamus. So I think we don't really know whether we can say that the thalamus is the gate. There is some evidence that the primary motor areas, the sensory areas, may be another gate, or maybe even more important. We just don't know. So that's one point. The other point is, it would be very important to study areas that are not the cortex or the hippocampus. In general, this hypothesis assumes that every circuit, independent of where the circuit is, every circuit that is plastic, that undergoes a lot of potentiation during wake needs the renormalization, because otherwise it will saturate. And, you know, it used to be that perhaps when I studied, we used to study the brainstem overall. You better not have too much plasticity in the respiratory systems, etc. But the reality is that I don't know now, there is very little evidence that there is any circuit that is not plastic. And if that is the case, then every system needs sleep. They might need it in different doses. You know, some systems like the hippocampus, again, because of the way their function and the way they are built, might need a much quicker plasticity. Every night they really need to reboot, basically. Other circuits may build up this need for near normalization after longer periods of time. But that would be my hypothesis, that that's why we say really that sleep is the price for plasticity. And so in the brain, any circuit that is plastic would need sleep. And the same would be through phylogeny. The implication of this would be that as long as a network, you know, I mentioned at the beginning now the latest evidence for the need for sleep and regulation of sleep, homeostatic regulation of sleep, is for Cassiopeia or for the Hydra. So these are diffuse network. They are not a centralized brain, but they are plastic, of course. They are capable of complex behavior. And I would argue that, well, perhaps then, you know, because of that, they need a process of renormalization. So that would be the implication, and that would be also the implication that then if this is really an essential function, once you don't have too much plasticity, you don't have need for sleep.
Thanks a lot. Thank you very much. It was a very inspiring reply. Thank you.
Okay, Professor Chiara.
Thanks a lot. First of all, thanks for the very interesting and inspiring talk, and in particular for the comment about neuroprosthetics. As a professor working on this topic, and I know pretty well the work that you mentioned is super cool, and I do believe that this link between neuroprosthetics and neuroscience is going to be more and more important for both sides. You know, I call it the tale of the two cities, like the Charles Dickens novel, because it's going to be a circular, as you mentioned. One question, more a curiosity. You mentioned that probably different areas like the cerebellum could have different learning mechanism and then different impact related to sleep deprivation. But how far we can go in understanding this for more complex task, if you go for more cognitive task or more complicated behaviors. So, I mean, if you think in the long term, if you want, going back to what also Ugo asked you before, if you want to make another jump in more complex task, what could be necessary in terms of changing maybe animal model or the way you are looking at the change in the plasticity?
Exactly. Well, I don't know. That's why I pointed to the neuroprosthetic learning, but it's still, you know, it's fantastic again because you have control. You choose which neuron is going to be causal, correct? You choose those two. You have total control. You know the system. But again, this is primary motor cortex, which is like the rotarod, the complex wheel. There is some other tasks, but they are in the striatum, but they are all motor tasks. There is one very nice model that is visual field, expansion of visual field, that's been used by the group of MIT, and that's the one in which they can show that the behavioral effect on the growth or shrinkage of the visual field, they can follow single visual neurons in V1, and actually then they can follow single synapses and follow the activity of some immediately genes. That, unfortunately, they haven't studied sleep in that, but that's another one in a different, on the perceptual level. In terms of declarative, so for instance, we are trying to probe higher order, if you wish, cortical areas, which it's already challenging in a rat. In mice, I learned, I was told don't even try, because indeed there is a huge difference between the mouse and the rat in terms of complexity of prefrontal cortices. But that's what we are trying to do, the forced choice, kind of tasks. But the problem is for us, when you go with electron microscopy, or even if you go to photon imaging, you basically need to know more or less where to look at. What kind of layer, what kind of neurons. And these tasks are not there in terms of characterization that we can then look at the effect of sleep. That's the bottleneck, I think, now, even more than the technology per se, is really the behavioral task that is limiting.
I understand. Thanks a lot.
Professor Faragona.
It's not my question, but there is an alumnus who is unable to ask questions because his kids are sleeping right now. So he wrote to me.
Sleep is important. I hope you remember this.
Yeah. We won't interfere with Francesco's kids. But the question was, is there any benefit in forcing sleep somehow, in extending sleep?
Well, it depends. So there is definitely benefits in banking sleep, meaning if you are fortunate enough that you can sleep in advance, knowing that you will be sleep deprived, you can nap or you can sleep more, that has been shown to help to reduce the cognitive impairment of sleep deprivation. Again, if you are able to do so. Prolonging sleep in general, if you just keep going, the late sleep seems to be shallow sleep. For instance, is not very rich in slow waves. And we think that because of that, perhaps a lot of this process of renormalization is not very efficient. And so, but I don't have any evidence, but even at the behavioral level, it seems that the benefits of sleep are not there. Instead, for the preventive sleep, they are there.
Francesco, thanking you. Thanks, Chiara.
Is there any other question? Okay, if it's not, I want to thank you, Chiara, too, for this seminar. And if I may, I want to do my own question. And I'm quite young, so I'm sorry in advance. It may be not probably on point, but I was asking to myself if is there any knowledge about the correlation between the sleep stages and this sleep function. I'm asking if REM sleep and non-REM sleep are functionally relevant for this.
We don't know. As I mentioned before, all what we have is total sleep. And so we don't know already. I would bet the REM has the same function as non-REM, but again, I have no evidence. And I am very skeptical of trying to, but this is my personal opinion, but I am very skeptical of trying to assign to different stages different function. And you have to remember also that, these stages, for instance, are very defined in humans. They don't really translate to animals like rodents, let alone to flies. And yet again, for instance, the effects of sleep deprivation in flies is very obvious. The evidence of renormalization. But you can't say that there is, the same staging of sleep in flies. So I tend to try to see what is common, and I tend to believe that, staging per se may not be the most crucial aspect.
Thank you. Thank you so much.
Sure.
Calogero, do you want to wrap it up?
Okay, I think it was a very nice debate, and I simply would like to thank again our invited speaker, and thanking her a lot, truly, for the time she committed to stay with us. Thanks again.
Thank you. And if you have any other question, just send me an email.
Grazie Chiara.
Buona serata, buonanotte.
Have a good night.
Buona giornata a te.
It's important, so be protective of your sleep.
We're going to sleep.
Ciao Chiara.
Ciao Paola.
Arrivederci.
Good night. Thank you, everyone.